So this is part 2 of chapter 5, and we left off discussing the polymerization of monomers. And so we were talking about amino acids and how that's a single monomer when you just look at one amino acid. But when you string them together, you get a polypeptide or a protein, which is the polymer of a protein. And the difference between a polypeptide and a protein, you can use them interchangeably for the most part, but a polypeptide is generally a much smaller um, protein. So it's not quite a protein yet. It's just a smaller chain of amino acids. And so, for example, we get a dipeptide. That means that would be just 2 amino acids long. And so again, a polypeptide is a smaller chain of amino acids, but the protein is actually when we get into the different levels of structure, forming. So the different primary, secondary, tertiary structure, when we actually go from our chain of amino acids into the actual final structure of a protein, which you need a longer series of amino acids to get. Okay, and the last monomer that we'll look at is a nucleotide, which is the building block for a nucleic acid. And so when we get a number of nucleotides to polymerize, then the polymer that we're making is a nucleic acid, such as DNA or RNA. And we'll look at each of these more individually in a bit here. So the first biomolecule we want to take a look at is sugars. Most names for sugar end with OSE, so like glucose, sucrose, lactose. And they're classified by the number of carbons that they have. Generally, the monomer of a sugar is going to be 3, 5, or 6 carbons. And so a 3-carbon sugar is a triose, a 5-carbon sugar is a pentose, and a 6-carbon sugar is a hexose. And the examples are given there. These are all, when they're just a single sugar molecule, that's called a monosaccharide. And a monosaccharide generally have molecular formulas that are some multiple of the unit CH2O. For example, we'll look here at the monosaccharide CH2O. Here our example is glucose. And it's a simple one monomer sugar. There are 2 things that all monosaccharides have, and that is a carbonyl group, so the C double bonded to an O, and multiple hydroxyl groups, or so OHs. And if you look at this glucose molecule, you can see how that occurs. But you may be wondering, where is the double bonded oxygen to the carbon? And that actually, you only will see that in the linear structure of the monosaccharide. So here we have, this is glucose here as well. And we can see that double bonded carbonyl group right there. But what happens, and we'll look at this a little more carefully in the next slides, but in liquids, most sugars are going to go from this linear structure into a ring structure. But before we look at that, let's talk about disaccharides. So disaccharides are when we have 2 monomers of sugar. So for example, glucose and glucose, or glucose and fructose. These will make up a disaccharide. And then a polysaccharide is when we have more than 2 monomers of sugar attached together. Okay, so moving on here. So looking at the sugar structure, 5 and 6 carbon sugars do form rings when they're in solution, so in liquids. So you can see here the linear form. And again, this is glucose that we're looking at. And you can see this is the process it goes through when it's put into a solution. We actually get well, first of all, before we can talk about what happens, another thing that we need to be aware of, I'm actually going to go to the next slide. So here's the next slide. What this is showing is the ring formation of the chain of carbons we have here, the hydrocarbon chain. And actually, it's more than hydrocarbons because we have some hydroxyls. But what we have here is an oxygen, and to the right of the oxygen, we start numbering our carbons in the ring. So carbon 1 is always going to be the first one to the right of the oxygen in the ring structure of a sugar molecule. And then we go clockwise to number the rest. And so here we have all the way up to 6. And the other thing that is shown in this slide is the fact that energy is stored in these carbon-to-carbon bonds. And that's important to remember, kind of looking at the bigger picture, that sugars or carbohydrates are the energy storage molecules in the human body and in all organisms, actually, beginning with producers, with plants, who are actually converting sunlight into chemical energy in the form of sugar. And so the sugar's actual storage of energy is actually in these carbon-carbon bonds. And that gets harvested in cellular respiration as those bonds are actually broken apart through a process in a series of chemical reactions. So going back here and looking at our linear structure of a sugar molecule, you can see the numbering system. So we have the 1 here, the 2, 3, 4, 5, and 6. And the oxygen that we start with before we start numbering is actually this blue one right here. So it's part of the hydroxyl group on the 5th carbon. So the glucose ring is formed when this molecule is placed in solution, and the oxygen on carbon number 5 links with the carbon that's part of the carbonyl group here on the end at carbon number 1. And so what happens is this hydrogen that's here on this hydroxyl for the 5th carbon actually gets transferred over to carbon 1 over here, to the oxygen over at carbon 1. To actually form a hydroxyl group. And then you can see here how now we get, so everything actually gets accounted for. So our carbonyl group was a C double bonded to an oxygen. That's broken so that that carbon can now bond with this oxygen here. So carbon still has its 4 bonds, 1 here, 2, 3, 4. And it's able to do that because the second valence of oxygen that was double bonded is now filled with the hydrogen that was part of this hydroxyl group. So that's essentially how the chemical explanation for what's going on with the ring structure, just looking at glucose. But that would go for all 5 and 6 carbon sugars. The same series of steps is actually what would be occurring. And that numbering system, again, of which carbon is which is important. And it's always going to be, when you look at your ring, that the first carbon after the oxygen to the right is number 1. So now, how do we build? We go from a single molecule of sugar, the monosaccharide, to a disaccharide or a polysaccharide. And that's going to occur through dehydration synthesis. So dehydration synthesis is the removal of a water molecule. And so these monomers get connected by covalent bonds, and the bond that's occurring between a monosaccharide and another is called a glycosidic linkage. And the dehydration occurs when one monomer Sorry about that. One monomer it gives up a hydroxyl group, an OH, and the other one that's bonding provides a hydrogen atom. And so in that way we get our hydrogen, our hydroxyl to form our H2Os. So that's where our water comes out and where we get the term dehydration synthesis. This is also known as condensation synthesis or a condensation reaction. So the same idea, condensation, you're pulling out a bit of water, a drop of water. And the same thing with dehydration. We're pulling out a drop of water, and that is going to be building a molecule. So the bond again that's formed is called a glycosidic linkage. We can also have the reverse process occur. So if we wanted to break apart this glycosidic linkage, the way we would do that is to add water to it. And so that process is called hydrolysis. And by providing that water molecule, we're basically just reversing this process to give back our hydroxyl and our hydrogen so that we end up back with our monomers. So now when we look at this problem here, we can start to think about the answer to this. So looking at what they're asking for, we have C6H12O6, so the formula for glucose. And the question is asking, what is going to be the molecular formula for a polymer that is made by forming 10 glycosidic linkages, so joining 10 glucose molecules together by dehydration reactions. And actually, I just caught myself here with a problem because the answer to this is actually going to occur by figuring out how many glycosidic linkages are you going to make if you're linking 10 molecules. And the correct answer is actually 9. So if you have 10 molecules, if you think about it, if you just draw out 10 circles and make bonds between each one, you're going to count up that you actually only need 9 bonds to link 10 molecules. So what you then need to figure out in order to do this problem, and you can pause this if you want to try and figure it out yourself, but I'll go ahead and continue to give you the answer here. So now we know that if we have 10 molecules being polymerized into a single chain, then we have 9 dehydration reactions occurring. So we have 9 water molecules that will need to be removed from this long polymer. So what we would do is we would say we have 10 of these molecules of glucose. So we'd say 10 times C6H12O6 would give us the total number of carbons, hydrogens, and oxygens that are going to be combining into the polymer. And then from that number, you would take out the 9 water molecules. So you would then say 9 times H2O to figure out how many hydrogens and how many oxygens you would need to subtract from your 10 glucose molecules. And when you do that, you can end up figuring out the answer here. So I'm not going to give you the answer right now. You can check with me in class to see if you got it right. But I'd like you to try and figure out following those steps. Okay, so polysaccharides is what we end up with after going through the polymerization process or the dehydration reactions to form a polymer made up of the monomers of saccharides, of sugars. So these cost energy to build, they're easily reversible, and they release energy when they are broken apart. So for that reason, they're great storage for energy. And we'll continue this in the next portion of